being an environmental microbiologist in training, and having no interest or background in medical microbiology, I took a position at the Children's Hospital of Colorado in the Department of Pediatrics, and mainly thinking about the human lung and in the context of chronic lung disease.
Can you get closer to the mic?
Closer. Got it. And so I was asked to speak about sort of thinking about our exposures, and I should say I have no personal disclosures, but I do receive funding for our lab. So the idea now is to talk about what's actually seen in the normal human lung. So I spend a lot of my time thinking about chronic lung disease, but that's, I don't think, the first place you want to start when thinking about lung exposure. And then thinking about, based on the exposures that are being talked about in the room, sort of what's the magnitude of that exposure. And so it'll sort of build well with prior talks. So we don't actually know much about the normal lung. You'll note that every time you see an HMP slide, they don't actually show the lung as one of the sites. And that's mainly because it was considered sterile until very recently. And so the conventional wisdom is that the normal human lung is a sterile environment, and therefore there wasn't a great need to actually study the microbiology of the lung. As it turns out, so it's outside, so it's a mucosal surface exposed to the environment. And so I think the conventional wisdom now is more that it is a continuously challenged site. And I would argue that it's actually a very low load site, so it's not intended to be heavily colonized. But I think under the normal sense, the lung actually has a community. And so I'll go through a little bit of data from other groups and some of ours from kids, just try to convince you of that. Hopefully it's not too hard. So the sort of the best study we have of the normal lung, by the way, all this happens in adults because you don't do the primary way these are samples are taken by bronchoscopy. And so that is taking a patient in an adult. It doesn't require general anesthesia, but you insert a tube into their lungs and you muck around and you squirt saline in, you take samples, and you do a lot of things that, while safe, do carry risks. And so we don't really get an opportunity to do this in the pediatric setting. And so this study has been published a couple years, but they did a very interesting approach in that they used multiple bronchoscopes so that they could do everything above the vocal cords with the first scope. And this is like paralyzing the vocal cords, all the procedures that you do to get ready to actually enter into the lungs. And then they removed that bronchoscope and they took a second scope to actually go into what they considered the sterile site. And they also did controls throughout, so they swabbed the tips of the scopes before and after, they washed the scopes out and looked at what was there. So it's very interesting. The one limitation is that they were only able to sample 6 individuals this way because it's just a very intensive sampling scheme. So the goal of the slide is not really for you to see the names. What I want to point out is that you have basically the controls. So these are the extraction controls, the washes from the bronchoscope, and then you have repeats of the 6 samples going from the upper airways down into the lower airways. And so the main point of this is that one, the nice thing is that there's very limited overlap here in the extraction controls with what is the predominant organisms in the lung. But one of the things that we worry about, especially in these lowish biomass environments, is that all the reagents have a background. And ideally, you would actually sort of calibrate the experiment to have reagents clean enough to see your sample without having to amplify or to have significant amplification. And this will be more clear in the next slide where I talk about the actual copy numbers, the ribosomal copy numbers that are actually present in the reagents. And so the main point of this is to show you that these top 3 things, all the heat and what's present in the normal lung, come from 3 basic groups: Streptococcus, Prevotella, and Bionella. So these are associated with mucosal surfaces and are we're heavily colonized with these groups. And this appears to be what is present in the normal lung. So this is sort of what is there, and it sort of makes sense that you would actually try to have something that you recognize as not being that bad in your lungs, because the airways are a very reactive site. And so then one of the things that we think about a lot is the load. And so in these normal adults, they're basically using a quantitative PCR measurement to measure the copy numbers of ribosomal RNA genes present. And so this is where it's somewhat unfortunate that they have about 1,000 copies in the background. So the reagent controls have about 1,000 copies. But you'll note as you go from the controls to the upper airways that it's not that big of a deal. So you have 3 to 4 orders of magnitude higher load in the airway samples, and the upper airway samples relative to controls. What's more problematic is that as you get into the lower airways, you're now bumping up against that detection limit. And so it was talked about earlier about sort of validation and understanding or validating the results. What's a negative result? All those things. So that's again something that we think about a lot more in the clinical side of things, because there's a huge regulatory framework if we ever do try to push these assays into a clinical setting. So right now this is all being done as a research test, and so there really aren't any specific requirements on validation, but we do spend a lot of time thinking about that. And that's also partially because my space is located inside a CLIA certified lab. So we're not under the CLIA certification, but we actually interact with people that spend a lot of their time doing the validation and assay development that's required to actually use this information to treat a patient. And so that's really one of the goals of this work in the CF world, in the chronic lung disease world, is to actually learn how to use these types of assays and apply them in the clinic. So just to link this, so now we are able to do something called sputum induction. So if you nebulize hypertonic saline into a human lung, it will cause you to cough. And since all of us produce mucus, it'll actually cause you to produce mucus that gives an adequate sampling of the lung. And so we have been able to do that on a very limited basis in adolescents. So these are basically teenagers of people very closely linked to the program, I think is the best way to describe it. Yeah. And so again, so this is work actually with longer Sanger reads, and so we again see that now we have Streptococcus mitis group. So there's a bunch of Streptococci that look very similar, even if you have a long read, and Prevotella magenta as the sort of 2 main things that are showing up in these healthy adolescents. What's interesting is if you can now do the same procedure on CF patients that are stable, so they're not. They have declined lung function, but it's their baseline lung function. We actually see very similar communities. And so what this suggests is that even if you have a chronic lung disease, when you're not in an acute period of disease, that you look pretty normal, which is somewhat interesting because one of our goals is actually to think about how you manage the ecology of the lung over the course of a decades-long disease so that you actually increase life. And so what this says is that they're actually fairly normal. What we don't understand, though, is that periodically they have what's called a pulmonary exacerbation. And this is where you'll lose 20 to 30% of your lung function, and it requires a hospitalization for typically about 2 weeks of IV antibiotics. And so there you see that we do see some indication of the normal organisms, but I've truncated this heat map. Once they exacerbate, you begin to see in individual patients very high amounts of individual things that aren't typically normal. And so what we really are trying to understand is what takes you from essentially a steady state, from sort of an engineering thought of this. I mean, if you think about the lungs as sort of a chemostat, what we really want to do is keep them in steady state as much as possible. And understanding this trigger that actually takes them out of exacerbation. I don't have answers for that. The other thing that we've spent some time looking at is we don't typically have bronchoscopy access to pediatric lungs. So in pediatrics, it requires general anesthesia. It's a much more complicated procedure, and so it's very limited. So what we tend to rely on are oropharyngeal swabs, so basically swabbing out the back of the throat, or sputum samples. And so what we've done is we've taken a small cohort and actually looked at how do those samples compare so that we understand sort of what we're able to learn from the different types of samples. And so the interesting thing here is that they actually look pretty similar. So we're actually obtaining very similar information So we see CF pathogens, so what we typically associate with CF lung disease as marked. One thing to note, that came up too early, but one thing to note is that some of these pathogens are clearly enriched in the airways. And so this is actually part of my model of sort of how this all works, is that the upper airways are really a significant source of what we see in our airways. And that once in the airway, there's definitely selection that goes on. So there's some things that are better at making it in the airways than others, including things that we perceive as pathogens, because that tends to be what we find when someone is sick. And then there's also the background of Prevotella staph, or Prevotella strep and Bionella. You'll also note that in the sputum samples, there's some small areas that come up. These are frank pathogens. So Chromobacter and Stenotrophomonas that are associated with CF lung disease, they only show up in the sputum. And so again, it's interesting that there is definitely selection in lungs. So if the upper airways are the source of a lot of what's there, how does it get there? And so pulmonary aspiration is something that's probably widely known. So it's any entrance of secretions from where they're not supposed to be in the lungs. So every time you try to breathe your drink, that's sort of an aspiration event. And so you have frank aspiration, which is sort of your typical, this is why they don't let you eat food before they put you under anesthesia for surgery, where you would actually get large volumes. That's a very bad outcome generally, because your lungs aren't really designed to have acidic fluid in them. But the more important thing, I think, is what's called silent aspiration or microaspiration. And so there's not very much data on how much aspiration that represents, but the best study, they actually applied during the sleep study, they applied colloidal sulfur compounds that were radioactively labeled, and they could actually determine a minimum estimate for the volumes that a normal human adult breathes in every night. And the answer is that you're taking in on the order of tens to hundreds of microliters. This was in 50% of the subjects. So it's not clear if only half of us aspirate or we only aspirate sporadically. The interesting thing is 2 of the 10 aspirated both nights of the study, whereas the other 3 aspirated only on a single night. But the main point here is that if you have very high loads of organisms in your upper airways and you're putting microliters of that into your lungs at night, that's a very significant source of exposure. And again, I think these are organisms that we've already decided ourselves. So if I'm going to put something into my lungs, I think that's probably a reasonably good thing to have. I think there's bad and less bad in the lung, and so these are very far over on the less bad scale. And so now to try to bring this more towards what really the topic of discussion is what are the sources of exposures? So we think a lot about bioaerosols. I think municipal water is something that should be thought about much more. Food's an interesting question. So I don't think you have to worry about the bag of Doritos as a significant exposure to microbes, but any kind of fresh fruit or produce certainly has some load on it. Again, what that load is what's not clear. So I've tried to find studies that actually use the same quantitative PCR assay to measure quantities in bioaerosols and food and water. It's not widely been done. So there is one, this is a dust study where they found 10 to the 3 to 10 to the 5 genome equivalents per cubic meter. And so I think that's probably on the high end of the load. Dust is basically a surface that can be colonized. We don't have good measurements for what is more typical bioaerosol, sort of an air sample cult. That what we do know is that they're very low biomass. We've actually run a few assays, just sort of single, not triple bit assays on bioaerosols, and there's very little DNA there. And again, when we look at sort of what we see in these bioaerosol samples, there's limited overlap with what's predominantly in the lung. So while it is a source of exposure, it seems that the host, the person's own upper airways, actually swamp that. Water has a much higher biological content. We've run just a few water samples, and that's where this 10 to the 7 and 10 to the 8 gene copies per liter come from. So there's a significant exposure. There's very limited overlap with what you see in bulk water versus what is in the airways, but we do see many, many opportunistic pathogens. So from the various studies that have happened in Norm's lab, all the CF pathogens are present in municipal water, shower heads, a lot of these environments where we're constantly exposed, but how they challenge, how they actually invade the host is the real question. And so for food, I wasn't able to find anything that measured it with this technique. But again, I think it really depends heavily on the food. So if you're, organic lettuce is probably a much bigger load than something that's heavily processed. And so I just put this back up to sort of give the context of the exposure. So, again, these are the loads that we're seeing as normal in human adults, and so air is down and sort of comparable to what we're seeing in the airways. Water is much higher, but I don't think liter is the right unit. So I think as an exposure source, it's probably more in the milliliters, and so that actually puts it down into this level. So the load is probably pretty equivalent to what's in the lungs, but it's got to get past a much higher and heavily colonized upper airway to have a productive invasion of the lower airways. And so I've been given the sign, so again, the model is really that the airways are continuous and that it's a difference in load. And so you're basically looking at the same things in the upper airways that we know are heavily colonized in the lower airways, and that the magnitude of exposure is probably much more yourself for getting things into the lower airways where you actually have to worry about what we would think of as a productive infection. And I think what we really want to understand is how you have a clinically relevant exposure. And with that, I'll take questions.
So I noticed a lot of the species or genera that you're talking about are really common in the oral biofilm. So you're talking Bonella, Prevotella, Streptococcus mitis. A couple of things come up. First, you have to go through that area to get your samples, right? So there's an issue of potential, contamination, making sure it's not the oral biofilm you're getting. But secondly, you said it was the human themselves are a major source of the bacteria, and the mouth is culturing these guys all the time in gum pockets. So it's possible it's just going directly from the mouth and the oral biofilm. And also, I'm just wondering if you're looking at levels of gum disease at all in these samples, and seeing if people with really nasty gum disease may actually have, differences in their lung. I mean, taking gum disease or anything into account, is that a good idea?
So we haven't incorporated dentistry into any of our CF studies. It is interesting to note that there can be significant dental. So you can basically develop gum abscesses that are, in the case I know about in particular, it was silent. So this was a patient going for transplant, and you get a very extensive workup before transplant, and they found these massive just pockets of infection all throughout the gums that weren't, that hadn't caused enough pain or discomfort for the patient to complain. Maybe because these patients are typically on 4 or 5 antibiotics continuously, so you can have sort of growth without. The issue of oral contamination, I think it depends on what you mean by that. So I view it as the lower airways contaminated by the upper airways. The one point to make is that for our studies where we look at BALs in kids, since it does require general anesthesia, they're intubated. And so we actually go through the intubation tube to obtain the samples. And so there's still a caveat of carry in with the intubation process, but we're actually seeing our sampling is cleaner than sort of in the adults where they just kind of run it, snake you through the nose.
The sputum though comes out through, right? Is that right?
But so when we look at BAL versus sputum, it's very similar. So we get very consistent communities from individual patients. One of the things that same study, we actually have baseline 1 month, 1 year, And in a lot of kids, they stay very similar across at least 12 months.
Just if you looked at the influence of the air temperature and humidity that is inhaled on any of these functions, because we voluntarily sleep at a much wider range of temperatures than we choose during waking period. Voluntarily 10 to 25, involuntarily both below and above those temperatures. So I've always wondered what the effect was on the organisms, on the breathing rate, and on our ability to deal with what we inhale. And the literature says that the upper airway is a perfect air conditioner, but those studies are like 15 minutes long.
Yeah, so sort of this question of exposure and what makes exposures productive, that's the area that we're starting to think about it. So we don't have any data looking at temperature, humidity, anything like that in exposure. Again, I think so the other piece of information from CF is that if you do an oropharyngeal swab as a screening visit for infants, that you're going to then bronch because you're concerned about an organism they have. About 30 to 50% of the time, you don't find that organism in the lung. So I think that you basically need to invade the upper airways. There's also really good data from sinuses and CF as being a source for lung infection. So I think it's the invasion of this upper airways that we really want to understand better, because once it's there, all these other sort of environmental factors that might influence it really do get leveled out a little more.
Last one. Yeah, you guys got to talk at the break. We're just jamming on time.